So this video is going to be starting with chapter 11. We're doing our final system for this semester. We're going to be starting the nervous system. Chapter 11 through 15 are all going to cover pieces of this topic. Chapter 11 is going to be like the overall thought behind the nervous system and the tissue itself. Chapter 12 is going to be about the central nervous system. 13 is going to be about the peripheral nervous system. And then 14 and 15 are going to be parts of it as well. So when we're talking about the nervous system, one thing to remember is that we are talking about a system that is the master controller and communication system inside the body. So we're going to talk a lot about electrical and chemical synapses again in this chapter, because that is how we're going to primarily have communication throughout our body. So looking at the overall with 11.1, and this is going to be the functions of the nervous system. So there's 3 primary functions of the nervous system, and they all kind of every function will come back to these 3 pieces. The first one is going to be sensory input. So we are picking up signals from our environment. This is going to be monitoring changes outside or inside the body. So if there's monitoring changes outside the body, this could be temperature change, this could be electrical change, this could be chemical change, whereas if we're inside the body, we could be registering those same things, but on the inside. So this is going to be just monitoring that change, that sensory input. The second function is going to be integration. And after we sense something, we then need to process, and after that, we then need to interpret that information. And after we process and interpret the information, then we need to make a decision. And so that will be this second function, is making the decision of what needs to happen. And then the third function of the nervous system is making that decision, is actually doing it. So that'll be the motor output. So we have the sensory input, then we're going to have the integration, and now for this motor output, this is going to be some sort of response. This is usually going to be from the effector organs, and an effector organ is going to be either a muscle or a gland that will do something. So these are the 3 overlapping functions of everything that we're going to talk about this semester. Sorry, this chapter. And this unit will kind of come back to these areas. So you can kind of see how you have information coming in. It goes to the brain to be processed, figure out what to do, and then it will actually do what it needs to do. So it's a 3-step system. So when we're looking at the nervous system as a whole, it is divided into 2 principal parts. So the easiest way to think about this is the nervous system overall is going to be the top Mac Daddy head. So the nervous system is responsible for communication, and it's the master control system. It will have 2 branches, the CNS and the PNS. So the CNS will stand for the central nervous system, and this CNS, central nervous system, will be the brain and the spinal cord. Those are the only 2 structures that make up the CNS. Under the PNS, this is the peripheral, so it's everything else but the brain and spinal cord. So this is the peripheral nervous system. So it'll actually be your cranial nerves and your spinal nerves. This is everything else outside of your brain and spinal cord. Inside our PNS, we're actually going to have a couple more divisions. But first of all, the primary reason you have your CNS is going to be, this is your primary integration center. This is where you will be processing the what you have received, the information, and then the what to do. It's the integration and it's the control center. Whereas your PNS will actually be primarily the incoming or outgoing information. So it's going to be most of the sensory or motor output, where the CNS will be the thing that will control or integrate everything. When we look at our PNS, it will branch even further. So there are 2 branches to your PNS, and those are going to be the sensory branch. So this is your periphery. You have the ability to sense. This, you will also then have the motor. As we've already talked about, sensory will be input into the nervous system. Motor will be output of the nervous system. As we're carrying information, this is going to be toward the CNS, and this will be away from the CNS. So basically what we're saying is if this is my brain and my spinal cord, this is my CNS. My peripheral is picking up information here, and it is sending it to my brain or my spinal cord. That would be the sensory part. And then you're also going to have your brain processing and deciding to do something, and that information will then go down to the same spot. That would be the motor. That's the output. So CNS is toward, motor is away. So one easy way to think this is that this is going to be what information this is from and where it is going to, kind of like an address. So this is from the receptors of the body, and this information is going toward to the CNS. Well, this motor output, same thing, from to. This is going from the CNS. This is going to the body, the effector organs of the body. So it's kind of a circular pathway with sensory coming in, motor going out. Sensory also has another name. This will be called the, as I spell it wrong, afferent pathway. So anything sensory will be called afferent, so A. Anything motor will be efferent. So sensory has to come first, so that would be an A. After you sense things, then you get the motor output. That would be the E. So afferent and efferent are going to be these two terms that we'll use a lot. Your motor output will split even further into chapters that will be 13 and 14. This will be your somatic nervous system and your autonomic nervous system. These we'll talk about a little further, but the two branches for this chapter to focus on are the CNS and the PNS, and how the peripheral has the sensory and the motor division. So this will cover these two primary divisions right here with your efferent and your afferent, and where information is going. So when we're looking at this somatic and this autonomic nervous system, as I said, we're going to talk about the autonomic nervous system. This is going to be chapter 13, I believe, but we are in the motor branch. We have already actually talked about one of these branches. So inside the motor branch, our somatic nervous system will actually be our skeletal muscles. So this will be coming from, this information will be coming from the CNS, and it will actually be headed to the skeletal muscles. So this we've already talked about because we've talked about the neuromuscular junction. This is going to be for our voluntary muscles. So this somatic nervous system we have already talked about. And so we're going to talk about autonomic nervous system in chapter 3. So that's something to know. Okay, real quick, we're just going to compare the autonomic to the nervous system so you can kind of see this branch. So this is just a redraw of what I drew, where you're going to have your nervous system, your CNS, and then your PNS. Your PNS will do sensory, motor. Motor will have somatic, autonomic, and your autonomic will actually split into sympathetic and parasympathetic. We will talk about that later as well. So this is the branch. But what this chart is doing is that it is actually saying that the connection between the central nervous system and the peripheral nervous system is a reciprocal relationship where information goes from sensory to be integrated back out to be motor. So it is a true reciprocal relationship there. Okay, so inside our nervous system, both CNS and PNS right now, inside our nervous system, we're going to have different types of cells. So those are going to be called neuroglia or neurons. So when we're talking about the first type of cell, we're going to talk about neuroglia. And neuroglia are small nerve cells in the central nerve or in the nervous system. And these are going to essentially be glue. And they will support other neurons. So neuroglia, glia will mean glue. These are glue for the neurons. So glue for the neurons with this neuroglia. This is the first cell type we're going to have. And then we're going to have the second cell type, which is going to be a neuron. And this is actually going to be the cell that transmits signals. This is the cell that is responsible for the communication and for the control. So the majority of the cells we'll talk about are going to be neurons, but these neuroglia are responsible for gluing those neurons and holding those together. So we're going to start with our neuroglia, and then we're going to go into our neurons. So when we're looking at our neuroglia, we're going to start with our CNS. So there are actually 6 types. This is not a typo. There are 6 types of neuroglia, whereas this only says 4, but this is saying that it's within the CNS. So there are 6 types of neuroglia total. 4 are in the CNS, 2 are in our PNS. So this is where we're starting. So we're going to start with our CNS. The 4 cell types of neuroglia in our CNS. The first type we're going to have is going to be an astrocyte. An astrocyte kind of looks like a star. It's going to be the most abundant type of neuroglia that you will have in your CNS. And it's going to be connected to your capillaries. And its function is mainly going to be to support and allow our neurons to do what they do best. And it will also connect the neuron to the blood supply so it can get all of the nutrients that it needs inside our body to allow it to do what it needs to do. So that's our astrocyte. That's the first type that we're going to have in our CNS. The second type of neuroglia we're going to have in our CNS is going to be a microglial cell. A microglial cell. And these are really small kind of cells. And their function is protection and maintaining the neuron health. So making sure that the neuron itself is super, super healthy. And the way that it will do this is this will actually act as a macrophage. So it will destroy things that it needs to destroy. So with these, jump on down to, here's an example of our neuroglia, but here's our microglial cell. These are small, really tiny cells, and they are going to act as a macrophage, and they will clean and get rid of and phagocytize microorganisms in order to protect the health of the neuron. So here's what a microglial cell looks like. We're just going to jump on down and do it this way. The third one is the epididymal cell. So this is the third type of neuroglial cell in our CNS. It's usually going to be a squamous or a columnar type cell, and it may be ciliated, but what's important for this guy is where it is found. So it is usually going to be found where there are cavities where it needs to line. So the function of an epididymal cell is it will line parts of our nervous system to line cavities along the brain and the spinal cord. So it will create our first barrier between our 2 external and internal environments. And really what it does is it will do it between 2 different fluids. So in our nervous system, we do have our central, our cerebrospinal fluid. And our cerebral spinal fluid we'll talk about in chapter 12. Circulates, it's a liquid, it's water-based with a bunch of ions in it, and it actually circulates around our entire brain and our entire spinal cord. Well, what the CSF does is it's a fluid, but it actually separates the space between the cerebral spinal fluid and the extracellular fluid. So just the fluid that is around the outside of the cell. So this epididymal cell will actually create the space between these two fluids. So it's a barrier. So that's important. So you can see how this is the fluid-filled cavity. So then the fourth type of neuroglial cell that you have in your CNS is going to be called an oligodendrocyte. An oligodendrocyte is a branching, it kind of looks like an astrocyte. It will actually branch a little bit less, a little bit fewer. And what's really important about the oligodendrocyte is its function. And it will actually wrap around the nerves of the CNS. So it will completely wrap around, and when it does that, it will act as insulation. So if you were to take a wire that is in your house, an electrical wire, it is usually going to be covered in some sort of insulation or protection in order that that electricity stays where it needs in the wire. So these oligodendrocytes will essentially wrap around the nerves and create this same barrier of insulation. This insulation or this barrier is actually going to have a name, and that is called the myelin sheath. So as we start talking about this, we'll talk about it a couple times in this chapter. A myelin sheath is insulation that is wrapped around the neuron. So that's what an oligodendrocyte does. So those were the 4 types of cells within our CNS, but I said there were 6 types of neuroglia total, because that is, we have 2 types of neuroglia in our PNS. So these 2 types of neuroglia in our PNS are similar to our CNS. So these 2 types are, first one is going to be a satellite cell. A satellite cell. So this is in our peripheral nervous system. So this is in our sensory or our motor. This is not a brain or spinal cord. So a satellite cell is also going to surround and act like a neuron cell body in the PNS. So when we're looking at this one, he is going to act with the same function as an astrocyte in our CNS. So he is going to act like our neuroglia type 1, which was our astrocyte. So they're very, very similar. They're going to act as supporting our neurons, connecting to blood supply, but this will be a satellite cell in our nervous system. And then the second type of cell is going to be a Schwann cell. And a Schwann cell is actually going to have a similar function as the oligodendrocytes in our CNS. So similar to the function of, it was type number 4 that we talked about, of oligodendrocytes in our CNS. So what that means is that these will actually surround the fibers, and they will also act as insulation. So we're going to have our Schwann cells that are insulation in our PNS, and we're going to have our oligodendrocytes, which are insulation in our CNS. So that is the difference between the two. So there's technically 4 different functions of neuroglia. 2 of the functions show up in the CNS, 2 of those show up in the PNS, but they just act and have different names. So that's the neuroglia. Now we're going to kind of move to the bigger part of the cell that will primarily be found in your nervous system, and that's going to be a neuron. So neurons are the structural unit of the nervous system. They are very important, and everything we talk about will come back to a neuron. You'll also see it called a nerve cell. These are highly specialized, and they are highly specialized to conduct messages. Those messages may be called impulses or communication, but they're going to conduct messages from one part of the body to the other, and those might be called impulses. A couple special characteristics of these types of neurons that are super interesting. One is that they have extreme longevity. They will last a person's lifetime. So that being said, is you essentially are going to have the same neurons when you were 15 that you're going to have when you're 75. These neurons can last for a lifetime, which is great. Excitability basically means that it's going to have the ability to receive impulses, receive communication, and have electrical change. So all of the pieces we talked about in chapter 9 with the muscle system will come up to play again in the nervous system. A couple other features of a neuron. They're amitotic with only a few exceptions. Basically, this means that they do not have the ability to divide. If they do not have the ability to divide, we can't replicate and repair. We can't replace. So this is why brain damage, spinal cord damage, any kind of damage that occurs to the nervous system is super hard to heal, because our neurons, our primary unit of our nervous system, does not really have the ability to heal. So that's a big, big part. There are some exceptions. Your olfactory system, your smell, and your memory in your hippocampus have the ability that we know of to create new neurons, but in general, you do not create, kind of as a rule of thumb, you don't create new neurons. So that's an area of research right now, which is very, very cool. And with our neurons, they're going to do a lot of things in our body. So they have a high metabolic rate. They're going to need constant supply of O2 and glucose. Oxygen and glucose are going to be really, really important, and they need a constant supply of all of these. Okay, so when we're talking about our neurons, we're going to talk about the general structure. So we're going to start with what is called the neuron cell body. So the neuron cell body is going to look very similar to just what we called a cell when we talked about general composite cell. This may also be called a soma or a prokaryon. It depends on what kind of text you're reading or what type of information that you are reading, but neuron cell body may also be called these two. And this is where you're actually going to have the biosynthetic center of the neuron. This is where you're going to have all of the metabolism occurring. It will be doing all of its synthesis, all of its proteins, your rough ER, everything will be found here. It usually does contain a spherical nucleus with a nucleolus. So when you're looking at your neuron cell body, this is the general structure that you'll kind of see. Most people kind of draw it on a triangle, and you'll see why as we keep going. And then this middle part would be the nucleus inside our neuron cell body. Most of these cell bodies are going to be found in our central nervous system. So when we're talking about this cell body, most of them will be found in the CNS. If you put a bunch of these guys together and cluster these cell bodies together, and this occurs in the CNS, these guys are going to be called nuclei. So not to be confused with the nucleus, which is this, but if you put a bunch of these together, but they do it inside the central nervous system, they're called it's a nuclei. Whereas if you put all of these guys together and you kind of lump them all together, all of these cell bodies, but you do this in the PNS, it's not called nuclei. These are going to be called ganglia. So either the term nuclei or ganglia, what we're talking about is clusters of the cell bodies. It's just where they reside. So that's a cool, interesting concept that we'll hear a couple times. Okay, so our neurons are not just the cell body. There is also going to be something called neuron processes. And these are going to be branches or arms that will actually extend from the cell body. So both the CNS neurons and the PNS neurons will have this type of arm-like process that will extend from the body. And these processes are going to have two names. So if we are going to have all of these arm branches that are sticking out in our CNS neurons, these will be called tracts. All of these together will be called tracts. Whereas if we have all of these neuron processes in our PNS, we will call these nerves. So if you guys have heard of a sciatic nerve, that is what we're talking about here. That is a tract, or it's a collection of neuron processes that is in your peripheral nervous system, specifically running down the back of your leg. So that's where when you hear the term nerve, you're usually talking about a nerve that's in the core, arms or legs, neck or head, but not in the CNS itself. There are two types of these processes that will come off of your cell body. So these two types of processes are going to be called, you will have a dendrite and you will have an axon. So these are the two types of processes that will be coming off of the cell body. So when you look at the motor neuron here, you can kind of see I'll draw my motor neurons like this. This is the soma, or my cell body, and I will have all of these things sticking out the top. These will be my dendrites. So this will be my general kind of rule of thumb when I'm drawing this. So what are the difference between these two things that are coming off of our body? So to start with, we're going to do dendrites. So dendrites are going to be short branching extension, but there's lots of these branches. So these dendrites are actually really, really important because their function is that they are the main reception area. So these dendrites pick up the signal. So the signal is going to come into the body. They are also called the input region. So if this is my cell body and I've got all of these little branching dendrites, that means we are bringing information to them, and that information is going into the cell body itself. This is your antenna. If you've ever had an old-fashioned TV, these are the bunny ears. So this is the input or the receiving, and they will, as I said, convey messages toward the cell body. So these are short distance signals. So they will pick up signal and then they will direct it towards the cell body. These are going to send information toward the cell. This is one direction. So you can see here's an image where you can see what we're talking about with the cell body and then all the branches. So if that's the dendrite, our second type is going to be our axon. And we're going to talk about both of these a whole lot more. So like I said, in dendrites, there was going to be a lot per cell. This one, there'll only be one axon per neuron. So if this is my cell body, here's my bunny ears, my receiving signal, and here's my axon. We have already said that my dendrite is picking up signals and sending it to the inside of my cell body. But what is happening with your axon is each one of these axons, and they could be very, very, very long. It could be up to 1 meter long. And what they are actually going to do is it is the conducting region of the neuron. So this is the conducting region. If the dendrites are the receiving region, these are the conducting region. And essentially what that means is that it is going to send the nerve signal away from the cell body. So with this, what it's doing is it's picking up this signal and it is going to send it down the length of the axon, and it's going to send the signal away from the cell body. So you'll notice that on my drawing, I drew all the arrows in the same direction because neurons are a one-direction communication. They do not go backwards. There is not this direction. That does not exist. All communication that happens in a neuron will be from the dendrites to the cell body to the axon. It is a one direction flow of communication. And as this is one direction flow of communication inside the neuron, in our bodies, we will have these neurons connecting to other neurons. And as they connect to other neurons, this is where we will have connection points that are called an axon terminal, where we're going to secrete neurotransmitters and we're going to connect to another neuron and excite it or inhibit it. So we're going to talk a lot about this in the second video for this chapter. But just to get an idea that our neuron is one directional, it sends our communication down the length of the neuron, and it will eventually communicate with another neuron. So another thing about these axons that's really cool is we're not going to spend anterograde or retrograde, is that around this axon, there is going to be a myelin sheath. So as you're talking about the different types of glial cells, we had a myelin sheath in the peripheral nervous system and a myelin sheath in the central nervous system. So our axons will be surrounded by a myelin sheath. So this will actually surround the entire axon. Typically, we will break this rule in a little bit, but an axon will be surrounded by this myelin sheath. This myelin is going to be a fatty substance. So it is made up of like a fat and a water, and it will actually surround all of it. So the function of this myelin sheath is going to protect, and as we've mentioned before, will be responsible for insulation. And so it will help with the electrical insulation. It will help with the electrical signal that is going to be happening inside our nervous system. So these myelin sheaths. So we will have some when we're talking about these myelin sheaths. So here's my little neuron, dendrite. Here's my axon. My myelin sheath will cover this like this, and it will protect and it will act as insulation. When this happens, this is going to be called a myelinated fiber. Remember, fiber is what we're calling an axon or a process. There are some times in our nervous system where we will not have these, and these will be a non-myelinated fiber. And that is where you're going to have your dendrites, your one axon, and there is no myelin, and that will be a non-myelinated fiber. And this will occur a couple places very specifically inside your nervous system, and we'll talk about that. So when we are myelinated in our CNS, remember that is going to be an oligodendrocyte. But when we are myelinated in our PNS, that is going to be a Schwann cell. So that's from section 11.1. So our myelination in our PNS is going to be formed by Schwann cells. And the way I like to describe it is they call it like a jelly roll wrapping around it. I like to describe it more as a piece of tape that is surrounding something. So when it's actually surrounding, here is our neuron, and you can see how this is actually wrapped all the way around it. So it will wrap this way. So it's kind of like if you were to take a stick and wrap tape all the way around it, you're going to get this like folded, kind of rolled perspective. That is what your myelin is doing. It will act kind of, and it will look kind of like this. So you can see how it's wrapped all the way around. So that's what's happening with our myelination. There's one cool thing that will happen, and this is going to be called myelination gaps. They were called nodes of Ranvier. This is also seen in your book, and your lab manual still calls them nodes of Ranvier, and I will probably say that as well because that is what I am familiar with. But with our neuron, who looks like this with our dendrites and our axon, you may not see, and by may not, you will not see this entire axon covered in myelin like this. What you'll actually see is that your axon will be covered with sections. So it will be this, and then there'll be a blank, and then it'll be this, and then it will be this. So rather than taking the entire fiber and wrapping this fatty sheath or this tape all the way around it, it's essentially just doing sections at a time. When this happens, you'll notice that right here, there's a gap, and that will be called a myelin sheath gap, also known as a node of Ranvier, and that's really what I'm more familiar with, so I may accidentally say that. So when you do have a myelinated axon, you will also have this myelin sheath gap. If you don't have a myelinated axon and it's a non-myelinated fiber, then it doesn't matter. You're not going to have these gaps, which is really cool. So here you can actually see everything we've talked about. Here's our soma, or a cell body. We said that there was the ability to have lots of branching axons that are communicating over short distances that are the receiving signal. And then these are going to go down the one axon, and around our axon is going to be these little sections of our myelin. So this is our motor neuron. So when we're looking at these processes in our myelin sheaths, what we just talked about was that was our myelin in our PNS. Whereas if we have myelin in our CNS, remember this is our oligodendrocytes, you will still have the myelin sheath. So it follows the same trend that we just talked about. Okay, there's two other terms that you'll hear a lot as we talk about chapter 11 and chapter 12, and that will be white matter and gray matter. If you hear the term white matter, this is where you will have areas with myelinated fibers. Remember, we said myelin was a fatty substance. Fat has a white coloration to it. So that's where this white matter comes from, because the myelin is made up of a fat which has a white color to it. So you put a bunch of those fibers together, you get something called white matter. Whereas when you have gray matter, this is going to be areas without myelinated fibers. So if there's not the myelin around the axon, there's not a layer of fat. There is not a layer of white coloration. So gray matter is the color of the actual tissue itself. So you may have areas without myelinated fibers, or these would be non-myelinated fibers, but you may also have gray matter where it's just neuron cell bodies. And like we said, myelin will only cover axons. So if you have axons and fibers are the same thing. So if you don't have axons, but you have the cell body, you can't have myelin, therefore it is gray matter. So these terms will come up again. Okay, this last little section of chapter 11 is going to be focusing on how we classify the different types of neurons in our body. So when we actually classify neurons, we are going to classify them two ways. So we will classify neurons based on the structure, how they are formed, what makes their structure, and then we will classify them based on their function, what they do and how they do it. So just like everything else we've talked about, this will be our anatomy, our structure, and function will be our physiology. So just like everything else in this class, we're going to talk about the structure and function relationship, and we classify neurons that way. So we're going to start with the structural classification of a neuron. So with a structural classification, we are talking about the number of processes. So specifically for this will be the number of processes And these are going to be coming from the cell body. So remember, this is our soma, our middle part. So when we say the number of processes coming from the cell body, there are 3 types of processes. The first type you can have is unipolar. If you have something unipolar, you are going to have one process. If you have a bipolar neuron, you have 2 processes. And if you have a multipolar neuron, you are going to have multiple, lots of processes. So as a reminder, the types of processes we have are dendrites and axons. So here is our 2 different types of processes and our structural classification. So I started here first. So our unipolar, here is our cell body, and you'll notice it has one process coming off. It will still have its dendrites, look like this. So we're just going to actually do all of these in a color, so it's just easier to see. So we're going to do, all of our dendrites are in green, and you can see those there. And there still is our one axon. So it is still communication pathways in, up, and then down. So it is still, at the end of the day, one directional. It's just going to be more like a cul-de-sac type situation where you drive down the street, and to get to the house inside the cul-de-sac, you do have to turn into that cul-de-sac, and you have to turn out of that cul-de-sac and continue down the street. So there's only that one road to and from the house leading into the cul-de-sac. So that's kind of what a unipolar looks like. A bipolar will have 2. So here's our cell body, and there's going to be 2 processes. By definition, we will always have one axon. So if we only have 2 total, and we already have one axon because that's a definition, that means there's only one dendrite. So there's still all the branches. They are just not attached to the cell body. So we will have one dendrite attached to the cell body and one axon. That is a bipolar neuron. This is also still one directional, so you can see how information goes like this. Or you can have a multipolar neuron, where we're going to have our cell body, and in the cell body you see all of these dendrites that are coming off of it. That's a lot of processes. But then you're also still going to have the one axon coming off. And this is still going to be one direction communication pathway. So that's how you can do the structural classification of a neuron. It is based off of how many processes, dendrites and axons, are coming off of the cell body. Okay, so then the second type, here's a couple other examples so you can see. The second way we classify neurons is going to be the functional classification. And with the functional classification, I ran out of room for that one. I'm going to rewrite that. What we're doing with this is what type of information that this neuron is carrying. So it's not about how it's structured. It's going to be the direction of the impulse. So it's the direction, the communication will travel. So there's 2 different types. So you can have a sensory neuron. A sensory will also be called an afferent. This was covered at the very, very, very beginning. A sensory or an afferent neuron is going to be carrying impulses toward the CNS. So this will be from our receptors in our body, and this will be carrying information to the CNS. So that's the important part, because it's the direction of information. So a sensory or an afferent neuron is going to carry information to the CNS. Well, then we're going to have the second type, which is going to be motor. And this will be our motor neuron, also called an efferent. And this will be carrying information from the CNS. So it's already processed everything it needs to do and figured all that out. And it is carrying this to the effector organs in the body. So this will be our glands or our muscles. So this is carrying information away from our central nervous system. So the way I like to draw this is if this is my lovely little body and I were to stump my toe right here, my sensory or my afferent is going to carry information up. So that's to my CNS. This is my sensory. Whereas my motor or my efferent is going to send information from my brain or spinal cord out to my body. So that will be away. So this is essentially a pathway of direction. So one is to the CNS, one is away from the CNS. So there's a couple other features that are important about this, and we're going to talk about the structure within the function. So when we're talking about our sensory or our afferent neurons, the structure of these guys are going to be usually unipolar. So our sensory neurons will be almost all are going to be unipolar. And almost all of them are going to have the cell bodies in our peripheral nervous system. Or you could say the cell bodies are outside the CNS. So our sensory or our afferent neurons will typically be unipolar, meaning there's one process, and the cell body will be outside the brain or the spinal cord. Whereas when we have our motor neurons, the structure of these are going to be almost always multipolar, and these cell bodies will be in the CNS. So that's how you can actually tell the difference for these guys as well. So sensory and afferent is going to be unipolar with cell bodies outside the CNS. It's carrying information to the CNS, whereas the motor or the efferent neurons are going to be carrying information from the brain or spinal cord to the effector organs of the body. These are typically going to be considered multipolar with the cell bodies in the CNS. And then you have a third type, which is kind of an super easy type to remember, and this is going to be the interneurons. And an interneurons is essentially going to be the in-between neuron. So he will lie in between a sensory or a motor. So they are a between neuron, which means they can carry either sensory or motor information because they're just going to be the connection point between two. These are usually almost always in the CNS, and a lot of the neurons in the body are going to be considered interneurons because these are just the connection point between the two different types of cells. So you can see how we have our neuron types down here, so you can kind of see how those work. So that is the end of video A for chapter 11, 11.1 to 11.3. This video will actually also cover exercise 15 for lab, so it'll also be posted there as well. And this is going to be the primary, it's what exercise 15 is going to cover is the exact same information that's covered in 11.1 to 11.3.